Hello. Here we'll be doing meiosis, right? So let's see, what do we have. Meiosis, it's similar to mitosis. So if you have done mitosis, it will be much easier for you to grasp this. However, even if you didn't do it, you can understand. So the first thing I would like you to know that the difference between mitosis and meiosis. Okay? Over here, what happens is like we have this 2n cell. Let's say your body cells. Okay? They are 2n, right? But now from this one, we want to get gametes, which is 1n. So with the meiosis 2, we actually get 1n plus 1n, right? That's the thing we are getting. Okay, and then let's say this is your father. Okay, I mean this is from a male, from a father. Okay, right? And then a mother undergoes the same process. A mother undergoes the same process. Undergoes the same process in which this 2n and n divide to form. So you get sort of the same kind of thing here. Okay. Then what happens is when male and female mate, this one of the gamete from male goes and one of the gamete from female goes, so they sort of mix. Okay, so if they mix, they give you what? 2n. So it's very important that mitosis divide this to n, so that when you mix them again, you should get 2n. You shouldn't be getting 4n. Okay, so it's dividing first, and then this division number of chromosomes it joins and gives you 2n. You should know that humans have 46 chromosomes, right? They divide to give 33 and 33. So this n is actually 23. Okay, and even in female the same happens, and then this one goes with the female's one and gives you 46. Okay, and this is the child, right? This is the child. This is from the mother we get another 23. Okay, so that's the mechanism, or say the summary of this, whatever is going on over here. Now the only difference in the mechanism, the biggest difference, is that there is no replication in prophase 1. Here, no replication. You see, there is no replication. So what does that mean? That means that this gets divided. Okay, this gets divided without being replicated. So it gets the cell's chromosomes gets half of its original value, right? Means we know that in prophase, normally in mitosis, okay, normally in prophase there is a replication, right? And then in telophase there is division. So 2 becomes 4, and or 4 becomes 8, and then 8 divide to give you 4 and 4. That's normal case. But then in meiosis, the thing there is different case. Here there is no division in meiosis 1. Okay, in prophase. So what happens? There are 3 phases in meiosis. Normally they like to keep it in 3 phases. Okay, in prophase. We have three phases in prophase. The first stage is prophase as we know it. The second is metaphase. Third is anaphase and telophase. These are very fancy words, but then they are very simple if you just get the concept behind it. Okay, so I'll sort of summarize for you to understand it more clearly. So in the first thing, chromosomes pair up. Okay, chromosomes pair up, and this is called synapsis. Each pair is called a bivalent. Okay, so this is a very important point, that in the first step over here, what happens is that the chromosome pair up, right? Pair up. It's very important. Synapses. It's already written, but I'm just writing it down so that you can, note it somewhere. So here the main thing is chromosome pairing up. Okay, here, which is synapsis, in this first in middle prophase. Then the centrioles moving to the opposite ends of nucleus as in mitosis. Okay, so the centrioles are moving the same as mitosis. The centrioles normally move to come to the polar regions, right? They are together, but then they come all the way to the center here, right? They're coming here. Now in late prophase, what happens is another three processes happen, but then the one thing you should know, that this can be kept together. They're just divided because there is a distinction between them. Like when centrioles start moving, is really you can time up and see the different times at which this occurs, or this pairing up occurs, and these other things which we'll be mentioning now takes place. Okay, there's a time gap a bit, so we can distinguish them. We can keep them separately as middle prophase and late prophase. Now what happens in late prophase is, first a nuclear envelope breaks up, as in mitosis. Okay, a nuclear envelope breaks up, as you can see here. The nuclear envelope has broken up, right? That's the first thing. Nuclear envelope breaks up, and then crossing over of chromatids may occur. I should know that the first thing is synapsis, and here what happened? Crossing over. Here crossing over takes place. Okay, in the late prophase. We won't be mentioning all the things. I'll just be mentioning those very significant things. Okay, and nucleolus disappear as in mitosis. So this is the same, I think. Nucleolus disappear, nuclear envelope break up. Okay, this is normal. These three, this 1, 2, and the centriole movement, are common, even in mitosis are there. But the one which you should be noting down very carefully is the synapsis. You may be asked about this. It's the synapsis process, which is pairing up of chromosomes, and crossing over, which is exchange of materials between chromosomes. And how do that exchange of materials takes place? As we can see here, the chromatids, their chiasma sort of cross each other, and then the materials, this band is taken by this another one, and this one is taken by another one. Let me just show you clearly. Okay, you can see what happened here, right? This blue region belongs to sorry, maybe we can use this one. This blue region belongs to this one, right? Belongs to this chromosome. But then this one, as you can see here, okay, it's taken by, there is exchange of materials over here. Okay, this some of the blue was taken by this one, and some of the red was taken by the other one, right? You can see some difference over here. So this is the crossing over region. This region, you can look at it, and here is the place where crossing over takes place. Okay, having said that, we'll continue to the next step. Now hopefully this first step was very clear, right? And bivalent simply means two chromosomes, right? Bivalent showing crossing over, there are two chromosomes. Okay, bivalent means a pair of two chromosomes. Now we continue to metaphase. In metaphase, what happens is these bivalents line up across equator of spindle. So here when we talk about chromosomes, we are not talking about individual chromosomes. It's in mitosis. Here normally the chromosomes go together as bivalents, okay? They line up across equator of spindle attached by centromeres. Okay, so you can see here they line up at the equator. Now you should know that normally if this was mitosis, you could have only one, right? So there wouldn't be bivalence. There could be only one, because one there would be replication, or this would have replicated, and so they will be keeping themselves in a series, in a row. Okay, but here, since we have this occurring in bivalence, they sort of pair up before dividing in metaphase 1, right? So that was clear, hopefully. And then we go to the next steps, anaphase. Now here interesting things happen. Centromeres do not divide, okay, unlike mitosis. Here centromeres do not divide. Whole chromosomes move towards opposite ends of spindle. Centromeres first pulled by microtubules. Now normally we say in the other process, in the mitosis, okay, centromeres divide, but here centromeres do not divide. They stay in the same region in middle. I mean, they move along. These are the centromeres. Maybe I should just remind you what happens. Wait, you can see over here what happens in meiosis. Let me just show you anaphase here. Okay, you can see here these centromeres over here must have divided half and half, right? Centromeres must have divided, and this is in mitosis, and we will see also it is there in meiosis too. Okay, so we got our two chromosomes. Each pair of chromosomes divide, and each chromosomes go to the opposite end, right? And you should know that this string kind of thing are known as microtubules, okay? And these are the ones that this spindle apparatus. These are the ones that move the help to move this chromosomes to the centrioles in order to divide. Then in the last step is telophase, and you can see over here we have nuclear envelope reforming, and we have cytokinesis. Cytokinesis simply means that it is constriction. Now when this constriction happens, it is this same cell. There is sort of this constriction happening here. Okay, the same cell becomes this way. Okay, this is in the telophase. The constriction takes place, and then again this nucleolus, which was disappearing here in the prophase, this nucleolus is reforming, as you can see. nucleolus is reforming, right? And then the spindle remain over here, and chromosomes have reached poles of spindle. The chromosomes, as you can see, they started to move towards the pole, and here they have already reached the poles. Okay, they have already reached the poles. Now a nuclear envelope also develops. We said nuclear envelope here disappeared. You see this one, the nuclear envelope here, it started to reform again. So these four things, it is sort of like repairing. Okay, this is kind of like repairing. So hopefully that was clear. I'll continue in the next video. Thank you for listening.